Okay. Protein synthesis. Long video alert. I suspect this will be a long video. What is protein synthesis? Well, this is the real deal. This is the thing. This is how you make all the proteins and enzymes that make you. From DNA to protein. This is it. This is how we're doing it. A simplified version of it, but this is how we're doing it. So, what's involved here? The following things. So you need DNA. And remember, DNA is broken up into chromosomes. And then I always say the chromosomes have like genes on them. Genes. What are these genes? Well, one gene codes for a protein. A gene codes for a protein. So if all of your DNA is the book of you, and the chromosomes are chapters, the genes are the sentences. Sure. They code for the they're the thing. They code for the protein. And then the letters are A, T, C, G, right? The nucleotides, the DNA nucleotides. And now we're going to learn what the words are, because there are words. There's words. Those are called codons. There's words. There's a code. A code. You wanna be God, you gotta learn the code. Okay? Then you can make any protein, any enzyme that you want, and you can manipulate life this way. And that's what humans do. That's what we do. We can do that. The COVID vaccines are mRNA vaccines. So now you'll know what that means today. So we want to make a protein. The cell goes, hey, you know what? I need this protein. Let's make this protein. So what happens is a section of DNA that codes a gene, that has a gene for that protein, will be highlighted. There'll be something that highlights it. It could be a hormone. There are quite a few things. We'll highlight it. Not important. We're not covering that in Anatomy and Physiology 12. But just so you know, there's something that, and you can have things that highlight genes that you don't want. Chemicals and toxins that you ingest, certain chemicals and toxins can actually trigger unwanted DNA transcription, or protein synthesis. But anyways, so a section of this DNA is highlighted, and that piece of DNA, because DNA is kind of wound up inside, the nucleus. We don't want to make all the DNA. So that's why we have these things that highlight a piece of DNA, because we don't want to be making all the DNA all the time. That wouldn't be bad. Cells only make small portions of the DNA. So that section of the DNA is highlighted, and it's unwound from the histones that it's attached to. And then an enzyme, a magical enzyme, an amazing enzyme, one of the most well-studied enzymes of all time, called RNA Polymerase, an enzyme that basically every organism in the world has some version of RNA polymerase, and you can trace the evolution of life by the small differences in RNA polymerase that exist. Anyways, the RNA polymerase comes along and binds to the DNA segment at the start of this gene. And then what RNA polymerase does, remember when I talked about DNA replication, there was DNA helicase that split open the DNA, and then DNA polymerases that made the new strands. Well, here, RNA polymerase does it all. It does both. So RNA polymerase splits open the double helix, the DNA strand, unwinds and splits the helix. And what it does is it makes a complementary strand on one side from the 5 to the 3 end. We're not covering that in this. We're not covering the 5 theory. But anyways, on one side, except it's not complementary pairing DNA nucleotides. It's complementary pairing RNA nucleotides, hence the name RNA polymerase. It's making an RNA polymer. So that's the first step. So you're making a copy of the DNA now with RNA. And then it stitches those RNA nucleotides together. It stitches them together. and now you've got a copy of the gene, a complementary base pair, a negative copy, I guess, essentially, of the gene, a complementary base pair of the gene in RNA. And that RNA doesn't stick around. It doesn't hang out with the DNA. It breaks off. Those complementary pairs, they don't stick around. It breaks off. And now you've got that copy. You've got that mRNA molecule. It is free. It's just a segment of only the gene. In reality, that's what I tell you, but in reality, actually, it has a whole bunch of other stuff, and then before it leaves the nucleus, it's like modified and cut down so that it's actually the gene. But don't worry about any of that. It's just a copy of the gene. For us, we're just going to worry that it's the copy of the gene. And then that mRNA now is free. It can travel out of the nucleus because, remember, the DNA is trapped inside the nucleus. It cannot leave the nucleus, and so what good is it? It needs a messenger. And that's what that RNA is called. It's called messenger RNA. And so the messenger RNA, that copy now of the gene, is free to leave the nucleus via a nuclear pore and go into the cytoplasm, out of the nucleus into the cytoplasm of the cell. And in the cytoplasm of the cell are ribosomes. Remember, we learned about ribosomes when we learned about eukaryotic cells. And ribosomes are this combination of protein and RNA, ribosomal RNA, that are made inside the nucleus, those pieces, and they come together in the cytoplasm. And what happens is the RNA joins to the ribosome at the start of the RNA. It has what's called a start codon. We're almost ready to start talking about the code. I don't know how to get into the code. The code is complicated. Code makes students freak out, but don't worry. It attaches to the ribosome, and then there it is held in place. And remember, it's only half the strand. That mRNA is only half the strand, so the base pairs are exposed. And so it's attached now at the ribosome, and what happens is to this RNA come another RNA molecule called a tRNA, a transfer RNA. And the transfer RNA, one end of it has 3 base pairs, complementary base, 3 bases sticking out that are complementary to 3 bases on the mRNA molecule. The other end of the tRNA is attached to a big, juicy, wonderful amino acid. And remember, amino acids are the building blocks of proteins. So here the protein is starting to form. So the process of RNA polymerase turning DNA into mRNA, turning the gene into DNA, is called transcription. I don't want to mess it up. And then the process when in the cytoplasm, when the ribosome, when the mRNA is in the ribosome and the tRNAs are coming along and they're binding the 3 tRNAs that match the 3 nucleotide tRNAs that they're called an Anti-Codon that match the 3 nucleotides of an mRNA chain, which is called the 3 nucleotides of all the codon. They bring the amino acid, and then the next three nucleotides bases on the mRNA molecule bring down bind to another tRNA molecule, which brings another amino acid, and these amino acids then are stitched together. It's all happening on the ribosome, are stitched together. There's that strong peptide bond that forms, strong covalent bond that forms between the amino acids. This process is called translation. And so on and so on and so on. And so these amino acids get stitched together. They're attached to those tRNAs, and those tRNAs are attaching to and more amino acids. And you get, finally, a chain of amino acids. You get a peptide chain. The tRNAs break away as this thing is being made. As it's being made, this is called translation. It's called elongation as well. And then the tRNAs can actually go and attach to another amino acid. The same amino acid that those 3 letters, those 3 nucleotide, those 3 bases of the tRNA, they only match one amino acid. We're going to talk about that in a second. So that's the general gist here. And then the peptide is made and it can leave over there. And then there's a stop codon, and the amino acid chain now leaves, and it can do secondary. It gets formed in secondary protein structure or tertiary, and then tertiary, quaternary, and off it goes. And now it's an enzyme or a protein. Go do your thing. Go on, child. Go. So now let's break this stuff down. So let's look at some of it. So that's the overview. This is what we're talking about. Step 1, transcription. So here is a DNA molecule. You see a section of it. It's got RNA polymerase attached. So here's where the gene is. And RNA polymerase comes along. RNA nucleotides are base paired to the exposed. It unzips the DNA and then it base pairs the mRNA molecules, and it stitches them together. It forms those covalent bonds, it stitches them together, and the mRNA is made. And it also, RNA polymerase is so amazing, it also reforms those hydrogen bonds actually in the DNA molecule will reform themselves, right? Because they're poles basically. And so there it is. It's stitched together, and off it goes. And I have actually, look at this. Oh no, I've left my simulation running, and it's made a whole bunch of proteins. But here you can see, so here is DNA, here is a section of DNA, and what I'm going to do, don't worry about positive, so this is the thing that highlights the gene, so this is the thing that we need, this gene, and here is RNA polymerase. I'm going to drag an RNA polymerase, and it attaches, and look, it unzips, and it makes this mRNA, and there's the mRNA. And now the mRNA breaks off, and it's free to go. It's free to leave. So this being the nucleus, so this mRNA can leave the nucleus and run into a ribosome, and then there's translation, and the ribosome comes. This doesn't show you the details, but a protein is made that way. It's read by the ribosome. And look, it can be read more than once. It can be read on and on and on. If I leave this simulation running, we will make lots and lots of this protein, I'm sure. Let's go back. So, no. Okay, so RNA polymerase, so you saw that mRNA goes. Now it can leave the nucleus. So this is the part that gets crazy. So every 3 nucleotides, okay, of mRNA are called a codon, an mRNA codon. Every 3 nucleotides, right? I mean, they were originally in the DNA. So every 3 nucleotides. And they code, these 3 nucleotides, they code for a specific amino acid. These are the words. These are the words. The words code for an amino acid. So if you have an mRNA codon UUA, you will get leucine during translation. UUG, you will get leucine again. You can get the same one. AUG gives you methionine. AAA gives you lysine. And remember, there is a whole bunch of amino acids. Here is a codon chart that's in your workbook. There is also a handy one in here. There's a fun one in here, a codon chart. Now the codon chart, remember the codon chart refers to mRNA, not to the DNA and not to the tRNA. Students always think that this refers to the tRNA. No, mRNA. So, so that's the code, and there's some special codes. So, look, the DNA, 3 DNAs. Let's just look at it. If you want to get leucine, the DNA will have to have AAT, which then the complementary base pairs on the mRNA will be UUA. And then the tRNA, this doesn't show the tRNA, the tRNA would be AAU, and that tRNA will bring the amino acid leucine. Now notice there's U's and T's. Remember, DNA contains the base thymine. mRNA, instead of thymine, contains uracil. Do not ever put a T in when I ask you to do anything with mRNA, and do not ever put a U when you're asked to do anything with DNA. So remember, RNA, so tRNA, mRNA has a U instead of a T. Does the same thing, but it just to screw around with you. Some of these codons are special. So AUG is the start. So every gene starts with AUG, which means on the DNA it starts with TAC. So if you're reading the DNA, every gene starts with TAC, which also codes for methionine. So every peptide has methionine as the first amino acid. It might not make it. The peptide chain will have methionine, but then those peptide chains then have to be folded to give tertiary structure and whatever. They get modified later down the line. We saw that in the cell unit, that when they go to the Golgi body modifies and completes those peptide chains, so that methionine might get cleaved off. But the methionine, the AUG, lets RNA polymerase know this is where I start. This is the start. There are also stop codons, 3 stop codons, sorry. So on the DNA, ATT, ATC, UAA, UAG, UJE. Now it says here stop transcription of gene. Yes, it tells RNA polymerase to stop the transcription of gene. It also, during translation, these UAA, UAG, UGU tell ribosome that it's over. Translation is over. And basically how they do it, each one of those UAA, UAG, UGA codes for a tRNA that is not associated with an amino acid. It doesn't have an amino acid to it. So it means stop. There's a cute thing. It does this thing. It like curls back on itself. Don't worry about it. Okay? Things about the code. The code is known as being degenerate. Let me say that again. Degenerative, because you might have cut me off. This thing sometimes cuts me off. What does degenerative mean? Look at the code here for glycine. For example, glycine, the codons, so these are the mRNA codons, are GGU, GGC, GGA, GGG, which means that if there's a mutation or something goes wrong in transcription, if something happens to the first nucleotide or the first base pair, the first letter in the codon, basically, if it gets changed to a C, for example, you will get CGG, which is arginine. You'll get a totally different amino acid. That's bad. Oops. If you change the second one, let's say we change that one to a C, then we have GCG. GCG will get alanine. Bad. But if you change the third one, you will get glycine again. So as you go from left to right, the importance of the letter, of the base, goes down, degenerates. And overall, what that means is that you can have these mutations happen. Still, some of them might be in the first 2 letters. And for some amino acids, even if you change the second one, you'll still get the same amino acid, but not as often. When you change the third one, more often you'll get the same amino acid. So the importance goes down. It degenerates. That's what degenerative means. And this overall, numerically, what it does is it reduces the damage from mutations. And mutations happening all the time. Remember, we're these fields of energy, right? These fields of electromagnetic radiation, of electrons, and there's a positive zone and a negative zone, and then there's bonds where the electrons are flying together, atoms are bonded together by just a cloud of electrons, lumping them together, moving them together. Well, you are being bombarded by electromagnetic radiation from space constantly. About 3 or 4 times a second, I believe, is gamma rays are flying through. And usually they just pass through. They have so much energy, they just pass right past the molecules, right past everything. But sometimes they strike in the right place, right at the bond or right where they're not supposed to, and they will cause something to change, something to break. They will cause a nucleotide to go from being cytosine to being adenine, thymine, or uracil, or whatever. or while things are being put together, all of a sudden a cosmic ray screws it up. It's possible. Other things can screw it up. We're just these fields, these electromagnetic fields. So the code being degenerative lessens the impact of these mutations overall, numerically overall. Okay. Other thing to remember about the code is it's universal. All life on Earth uses this code. Any piece of DNA that codes for the mRNA that is CCC, for example, will give you proline in any organism on the Earth. It's kind of very solid evidence for us evolving from a single place, from a single source. There are some bacteria that have been recently discovered that have changed their genetic code. But they actually can do both. They can switch back and forth. But they've changed it basically because they've evolved a different code because they are under attack by viruses. And by screwing with the code, the viruses can't use their machinery, basically. The virus can't make itself, because what viruses do is they hijack this protein synthesis. So instead of making the protein that you want to make, you make viral proteins. This is how mRNA vaccines work. mRNA vaccines work in a manner, because what mRNA vaccines do is now that mRNA, instead of coming from the nucleus, comes from the vaccine. It doesn't do any of this. It's just an mRNA that enters the cell and becomes translated into viral protein. And the the cell doesn't know what to do with it. It'll just excrete it out of the cell. And so it's only one viral protein. So you're not making the virus. You're just making a tiny piece of the virus that doesn't function. It doesn't do anything. But your immune system then learns that viral protein structure. Well, what is this thing? I don't like it. And it learns it and it remembers it. And then next time you do get coronavirus, your immune system already recognizes it and associates it with something bad. And before corona can infiltrate a lot of your cells and start growing and dividing and establish itself and basically make you ill, start killing your cells in huge number, before that can happen, your immune system recognizes it, because if it didn't recognize it, it sort of ignores it for a while to figure out if it's good or bad. And by then it's too late. Then it has to mount a huge effort to try to get rid of it. And that effort is actually what ends up killing you, inflaming you, and giving you inflammation and fever. But here it recognizes it right away, and because the protein comes in, the enzyme, the virus comes in, and it has those proteins that the vaccine had made on its surface, and so your immune system just gets rid of it before it can establish itself. The mRNA vaccine is brilliant. Practice the code. Practice the code. There's a question in the review, and I will give you a worksheet, or there should be a worksheet, or I'll put a worksheet in somewhere in there. What else do we need to know? Oh, let's look at this tRNA. Let's talk about translation. So here is, look, a beautiful image of tRNA. So this is an image of translation. So here is the mRNA. There's the codons in the mRNA. And here are the tRNAs with the anti-codons. Okay, there's the anti-codons. And there they are. They're bringing the amino acid. A peptide bond is being formed. The peptides are being elongated. These two guys are bound. Peptide bond forms. This tRNA is already broken off from its amino acid, and it'll go off and it'll pick up the same amino acid. UCA will code for this exact same amino acid, so it'll come back. It can only have the same amino acid, and it'll come back, and then the next time there's an AGU in the codons, it will bring in that amino acid. Then this all happens at the ribosome. Here is an actual frickin' electron microscope image of an mRNA molecule being attacked by a whole ton of ribosomes. So, you see this ribosome here, but there's nothing to say that you can't have more than one ribosome attaching to the mRNA and making these amino acid nascent proteins. They're called peptide chains at this point, before they form all their tertiary structure or whatever. They're just nascent proteins. So if a cell needs a lot of this protein, it'll send in a whole bunch of ribosomes. We talked about it. There's that thing of ribosomes in the cytoplasm, or these can be embedded in the endoplasmic reticulum, the rough endoplasmic reticulum, and you make all of these proteins. So does that make sense? Have I covered protein synthesis? There's a good video here, protein synthesis by the Amoeba Sisters. Everybody loves the Amoeba Sisters. So remember, in the codon table, whatever codon table you ever get, these are not the tRNA anti-codons. These are the mRNA codons. Then they have a tRNA anti-codon that will bind to them and bring in this amino acid. No, tRNA is the unsung hero of this whole thing. Everybody always talks about an mRNA vaccine. Why not a tRNA vaccine? Well, it wouldn't work. And remember, there is always the start codon, AUG, which codes for methionine. And there are stop codons, which is reached on the mRNA. They do not correspond to a tRNA and then that makes translation stop. And I think that's essentially it. This is the thing. This is how you make all the stuff. Can I drag this away? It's in the wrong place. It's really, really bothering me. I don't know how it got there. Like, it's making me crazy. It's making my OC, if I had OCD, it's making my OCD go bonkers. Okay. There we go. Initiation, when the start codon binds to when it binds to the ribosome. Elongation as the peptide chain grows, and termination when you reach a stop codon. So there you go. Yeah, this is an amazing. This is your cells are constantly doing this. They're making tons and tons of proteins and enzymes, and this is how it's done. And, we have learned how to manipulate it because we can create now mRNA and send it into cells so it can make enzymes and proteins that we need, or maybe that we're making incorrectly. If there's a problem with your DNA, you can take injections of mRNA, for example, for insulin. Insulin's a big one if you are diabetic. Anyways, there you go. Protein synthesis.